Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. We are from Group 5. There are 4 of us. Firstly is me, Nur Ala Kainam Timanjadi, Nur Shazni binti Muhammad Suyud, Nurul Ain Izzati binti Syahri, and Nur Fatihah Idana binti Zairul. And today, we are going to present about Lab 6, which is growth study of microorganism in shape Flask. Alright, now I'm going to talk about the abstract part. There are 2 objectives that we have to achieve in this experiment, which are to collect experimental data on cell dry weight on the purpose medium of microbial experiment, and the second one is to calculate the mass doubling time from the microbial experiment. And generally, we use shake flask fermentation, but this time we use our method, which is by using yeast and sugar. There are 4 phases of microorganism growth, which are lag phase, exponential phase, stationary phase, and death phase. This experiment required us to use 20 gram, 40 gram, and 50 gram of sugar, along with 11 gram of selected microorganism, which is yeast, mix it with 100 milliliter of water for 15 hours. The microorganism in 50 gram of sugar of medium took the longest time to completely die with 18.5 hours, followed by medium with 40 gram of sugar and medium with 20 gram of sugar with 15 hours. And while the doubling time based on the maximum growth rate are 73.74 hours, 96.27 hours, and 51.73 hours respectively for the 20, 40, and 50 gram of sugar.
For the introduction, shake flask is a well-known vessel reaction that frequently been used for fermentation, screening, and process development of bioprocess engineering. So shake flask fermentation is the cheapest and simplest technique to grow microorganisms. In the fermentation of shake flask, Erlenmeyer flask is used for culturing microorganism. Culture microorganism can be done either in aerobic or anaerobic processes. However, shaken culture are usually applied for anaerobic process. Generally, filamentous microorganisms are grown which begin 1 to 3 days after inoculation and continues 3 to 4 days thereafter. The length of inoculation and fermentation time varies according to the type of microorganism, cultivation methods, and the type of desired metabolites. Due to the circumstances of limitation material and apparatus at home, we do some modification for this experiment that's suitable and safety to handle at home. These are minimum components that required in this experiment. First, carbon source, which is the carbon source that we use in this experiment is actually sugar to provide energy and carbon skeleton for the synthesis of various two other biological compounds. And number 2 is nitrogen source, which is purified water that we've been used in this experiment. And for the third is inoculum, which is we use is yeast.
A typical bacterial growth curve consists of 4 phases, including lag phase, exponential phase, stationary phase, and also death phase. During lag phase, the population of bacteria remain unchanged within certain amount of time. Thus, the gradient of bacteria over time, dx over dt, remain 0. The second phase is exponential phase. Within this phase, the growth pattern of the bacteria increase as the time increase. The rate of exponential, also known as doubling time of the bacterial population. The bacterial growth will reach maximum, which brings it to stationary phase. Bacterial growth reach maximum due to few limitations such as availability of space and nutrients. Lastly, the natural process of living occurs to the bacteria, which is death phase. The number of viable cell decrease at the time increase. This process shows the reverse growth compared to exponential phase. The dry weight cell can be calculated by the mass initial minus mass final. The rate of net microbial growth can be determined using the equation 1 over x dx over dt. Next, the doubling time can be expressed as ln 2 over the net microbial growth.
This is the material and apparatus that we've been used in this experiment, which is 100 ml of water, 11 gram of yeast, 20, 40 and 50 of sugar, plastic cups, weighting scale and sanitizer.
Prepare the materials for the experiment. Prepare 20 gram of sugar. Prepare 100 milliliter of water. Sanitize the cup used for the mixing process. Mix 20 gram of sugar with 100 milliliter of water in the sanitized cup. Stir it well until all sugar dissolve. Mix 11 gram of yeast with the sugar solution and mix it well. Set the timer at 50 minutes and record the weight every 50 minutes. Repeat the process with 40 gram and 50 gram of sugar. Record the changes in weight for 2 or 3 days.
For the comparison between 20 gram and 40 gram of sugar for the first few hours of experiment, 20 gram of sugar is forming more bubble compared to the 40 gram of sugar. This indicates that yeast in 20 gram of sugar is having cell growth higher compared to the yeast in the 40 gram of sugar. The result for the final hours of the experiment, we can see that 20 gram of sugar and 40 gram of sugar, there's no bubble exist in this cup, in this both cup, which indicates that the cell growth is become constant. For the comparison between 20 gram and 50 gram of sugar, for the first few hours of experiment, we can see that bubble form in this cup is very higher, and for the final hour of the experiment, we can see that after a few hours, there's no bubble anymore exist in both cup.
Next is the result of the experiment. For the first 20 gram of sugar, we use 100 milliliter of water, 11 gram of yeast, and 5 gram of cups. And what we can see here, at the initial time 0, the weight of the mixture is 134 until the weight of the yeast increased until it reached at the time 5.5 hour. The weight is 174 and it remained constant until the time is 6.5 hours and the weight remained to 174, and as the time increased, the weight of the yeast is decreased, which is at the time 15 hours, the weight of the mixture is 148. Next, it shows that the result of 40 gram of sugar, the water volume that we use for this part is also 100 milliliter of water, 11 gram of yeast, and 10 gram of cup. At the initial time 0, the weight of the mixture is 155 grams. At the weight continued to increase until it reached time 6 hours. At the 6 hours, the weight of the mixture is remain constant until the time 9 hours. At 9 hours and above, until it reached 15 hours, the weight of the mixture is also decreased to 166. Right now, we can see the result of 50 gram of sugar. For this part, we use 100 milliliter of water, 11 gram of yeast, and 5 gram of cup. At the initial time 0, the weight of the mixture is 157. As the time increased, the weight also increased. As what you can see here, at the time 9 hours, the weight of the mixture is 215, and it remained constant until it reached the time 13 hours, which is also 215 grams of the mixture. When the time increased, the weight continued to decrease, which is when at the time 18.5 hours, the weight of the mixture is 187.
So from the data tabulated from the experiment, we can calculate based on this table. This is for the medium of 20 gram of sugar with yeast. So we can calculate cell dry weight, cell mass concentration X, ln X divided by X initial, and the net growth rate cell for cell. So this is a sample of calculation for the data in the table earlier. So first we calculate for cell dry weight, which is the final mass minus initial mass. For 0 hours, cell dry weight we got is 129 gram, which is 134 gram minus 5 gram, which is the weight of the cup. And second, we calculate for the concentrations of cell, which is the cell dry weight divided by volume of sample. So for 0 hour, we calculate 129 gram divided by 0.1 liter, we got 1290 gram per liter. Third, we calculate for the ln X over X initial, and then we got 0. Fourth, from the linear equations, from the graph of ln X over X initial versus time, we got for the maximum rate growth of cell, which is 0.0094 per hour. And then fifth, we calculate for the net growth rate for the cell, which is the ln x2 minus ln x1 divided by t2 minus t1, and then we got 0.0154 per hour for the times of 0.5 hour until 1 hour. And then the last step is we calculate for the doubling time, which is we calculate ln 2 divided by the maximum net growth rate of the cell, and then we got 73.74 hour. So this is the graph that we plotted after we calculated using the calculation earlier. So the graph at the top is the graph ln X over X initial versus time, and the graph at the bottom is the graph of cell mass concentrations against time. So for the graph ln X over X initial versus time, we can see that there is a linear line and there's a linear equations given. So from the slope at the equations, we can get for the maximum growth rate for the cell. And we also can identify the phase for the cell. So this is the table for the data calculated for the mediums of 40 gram of sugar with yeast. So this is the calculations for the medium of 40 gram of sugar with yeast using the same formula as before. First, we calculate for the cell dry weight. Second, concentration of cell. Third, we calculate for ln X over X initial. Fourth, we find the slope from the linear equations from the graph of ln X over X initial against time. Fifth, we calculate for the net growth rate for the cell. And then lastly, we calculate for the doubling time for the experiment. So this is the graph plotted from the calculations earlier. So at the top is the graph of the ln X over X initial versus time, and then the graph at the bottom is the cell mass concentrations versus time. So from the graph of ln X over X initial versus time, we can see that there's a slope, and then from the slope, we can get the maximum growth rate for the cell, which is 0.0072 per hour. So this is the data tabulated for the calculations of a medium for 50 gram of sugar and yeast. So the formula used for the calculations for the medium of 50 gram of sugar with yeast is the same as the previous calculation used with the mediums of 20 gram of sugar and 40 gram of sugar with yeast. So this is the graph plotted after the calculations for the medium of 50 gram of sugar with yeast. So from the graph at the top is the ln x of x initial versus time, which is we can get the maximum growth rate of the cell from the slope of the equations, which is 0.0134 per hour. And then for the graph at the bottom is the cell mass concentration versus time. So for the discussions, the objective of the experiment is to determine the experimental data on cell dry weight for the proposed medium, which has been calculated at the calculation part. And second is to calculate the mass doubling for the microbial experiments, which also has been calculated in the calculation part. This is the graph plotted for the ln x over x naught versus time for the medium of 20 gram of sugar with yeast. We can see that there is a 4-phase happens in the graph, which is lag phase, exponential phase, stationary phase, and death phase. For lag phase, it happened from 0 hour until 1.5 hour with a rate of microbial growth net with 0.0077. The lag phase happened because the microorganisms are adapting themselves to the new conditions such as pH, temperature, or oxygen availability. Next is the exponential phase. It happened from 1.5 hour until 5 hours with the rate of 0.0615. The exponential phase, or known as log phase, happened because the microorganisms are growing rapidly and dividing to produce more generations. The growth rate of bacteria increases as time increases. Next is the stationary phase. It happens at 5 hours until 6.5 hours with the rate of 0 per hour. Stationary phase happen because there is no cell division in bacteria and the bacterial growth reach its maximum because of certain limitations such as space, nutrients, and that is needed for the growth process. The number of cells being produced is equal to the number of dying cells, and the waste from the bacteria also starts to accumulate. And lastly is the death phase. That phase happens at 6.5 hours until 15 hours with the rate of 0 per hour. The death phase happened because there is no available nutrients and the number of dying cells keep increasing. The number of bacteria decreases within time where there is a decline shows in the graph. Next, we calculate the doubling time using the formula of ln 2 divided by the maximum growth rate for the cell. The maximum growth rate for the cells, we can get it from the equations, linear equations in the graph, which is 0.0094, and then we substitute in the formula ln 2 divided by 0.0094, and then we got the doubling time is 73.74 hours. So this is the graph for the ln x over x initial versus time for the medium of 40 gram of sugar with yeast. So we can see that the lag phase happened from 0 hour until 0.5 hour with the rate of 0 per hour. For the exponential phase, it happened from 0.5 hour until 5.5 hour with the rate of 0.0542 per hour. For the stationary phase, it happened from 5.5 hour until 9 hour with the rate of 0 per hour. For the death phase, it happened from 9 hour until 15 hour with the rate of negative 0.0095 per hour. For the doubling time, we got the slope from the equations in the graph, which is 0.0072. We substitute into the formula and we got 96.27 hour. This is the graph of ln x over x naught versus time for the medium of 50 gram of sugar with yeast. We can see from the graph that there's also 4 phases, which is lag phase, exponential phase, stationary phase, and death phase. For the lag phase, it happened from 0 hour until 1.5 hour with a rate of 0.0066 per hour. For the exponential, it happens from 1.5 hour until 7 hour with the rate of 0.0293 per hour. For stationary phase, it happened from 7 hour until 13 hour with the rate of 0 per hour. For the lastly, which is death phase, it happened from 13 hour until 18.5 hour with the rate of negative 0.0095 per hour. The maximum growth rate of cell that is determined from the slope of the linear equations from the graph, which is 0.0134, is used to calculate for the doubling time, which is 12 divided by 0.0134, and then we got 51.73 hours. From the data obtained from the three mediums with different mass of sugar, the microorganisms in 50 grams of sugar took the longest time to completely die with 18.5 hours, followed by medium with 40 grams of sugar and medium with 20 grams of sugar, both with die within 15 hours. There might be error in the experiment caused by the human error when tabulating the data for the cell dry weight or when weighting the weight because of the long period taken to carry out the experiment and the lack of the apparatus. These experiments also is quite difficult to repeat because of the long period taken for this experiment. The mode of growing state for this experiment is static mode growth due to the lack of apparatus, and the medium goes through the phase without any mixing or shaking. Thus, the objective of the experiments have been achieved and the experiment have been carried out successfully.
Next, for the conclusion. In conclusion, based on the conducted experiment, the data obtained from three of the mediums with different mass of sugar, which are 20 gram, 40 gram, and 50 grams, the microorganisms in 50 grams of sugar of medium took the longest time to completely die with 18.5 hours, followed by medium with 40 grams of sugar and medium with 20 grams of sugar with 15 hours.
Recommendation: first, before start the experiment, make sure the apparatus must be wiped with alcohol, for example by using sanitizer. Second is the surface of cup should immediately close during preparation of inoculum to avoid contamination and uncontrolled factor. Third is avoid breathing in front of sample experiment while checking measurement and investigation. Fourth, time observation should be extended at least until 3 days to see reaction on cell growth. 5. Do not handle this experiment without wearing gloves and masks to obtain reliable data on cell growth.